I would like to thank the iMeta platform for this opportunity to present our research on the molecular recognition mechanism of microbial regulation of parasitic plants using Orobanche cumana as an example, and the related results published in the iMeta journal. The plant we are studying is O. cumana, a parasitic plant that only parasitizes sunflower roots. Known to infect arid and semi-arid regions, O. cumana is primarily present in Inner Mongolia, China. Other affected areas include the Mediterranean, Europe and Asia. Induced by the sunflower rhizosphere secretion strigolactones, the seed of O. cumana germinates and produces terminal haustorium tissue, which can connect with the vascular system of sunflower roots, absorb water and nutrients from them, and grow. Short growth cycle, light and small seeds, fast reproduction, seeds can lie dormant for 5 to 10 years. Presently, chemical pesticides, biological agents, crop rotation, and manual uprooting are the main prevention methods. The effectiveness of these methods is limited due to the parasitic property of O. cumana, which already damages sunflower before it emerges from the soil. In order to investigate the effects of microorganisms on O. cumana, we classified them according to the number of parasitized O. cumana as groups H, PL, PM, and PS. We found that the rhizosphere microbial community structure differed significantly based on amplicon analysis. Xanthomonadaceae relative abundance increased significantly with parasitism, while Microbial showed a decrease. In the regression model, the number of O. cumana was significantly and positively correlated with the abundance of Xanthomonadaceae as well as with CEC and CECK content. H and PS group macrogenomic sequencing and construction of co-occurrence network reflected that the microbial communities are more tightly bound and complex in healthy plants. Previous studies have shown that the more complex the microbial community is, the more stable it is and the stronger its ability to resist invasion by foreign bacteria. Sunflower rhizosphere microbial resistance to O. cumana parasitism is similar to this theory. The KO pathway and enzymes were corresponded to the top 10 bacteria in the KGG database, and the results showed common clustering to 3 genera, Lysobacter, Varioverax, Pseudomonas, of which Lysobacter, Varioverax, the 2 genera with higher abundance in PS, are reported to have with antibiotic-producing, cellulose-degrading effects. And Pseudomonas is the one with higher abundance in H, which is associated with the existing reports of O. cumana stem rot, and inhibition of parasitism. Could these bacteria influence O. cumana's germination and parasitism? Using the traditional bacterial isolation and culture method, we collected 5 strains. O. cumana germination experiments were carried out using these 5 bacteria strains. HX79 not only increased the length of the shoot tube but also promoted O. cumana germination. The HX79 fermentation solution was added 1 month before planting sunflowers. The final results showed a significant decrease in sunflower emergence rate and a significant decrease in parasites on the root system. O. cumana parasitism was also inhibited by Pseudomonas aeruginosa HX1. We screened the metabolites of HX79 bacteria for molecular model prediction. Using OCKI2D2 reaction model, we screened 3 compounds, cyclic dipeptide, 2-deoxyadenosine and 2-hydroxyadenosine, to take in seed germination experiments, and the results showed that all had the effect of promoting the germination of O. cumana at the optimum concentration of 10 To investigate the mechanism controlling the parasitism of O. cumana, we performed integrated multi-omics analysis in this study. It was demonstrated that rhizosphere microbes have a regulatory effect on the parasitism of O. cumana. By constructing a molecular binding model, we successfully predicted 3 compounds that promote the germination of O. cumana and clarified their molecular recognition mechanisms. For practical purpose, the strategy of screen compounds appears applicable to other parasitic weeds, including Orobanche species. iMeta, a journal to change the world.